Model systems link genetic determinants to the Generative Toolkit. The generative and ecological phenotypic effects of genes and alleles should not be viewed in isolation. Most aspects of development depend on complex interactions between these elements. Unfortunately, one legacy of the modern synthesis was the tendency to analyze speciation in terms of changes in the frequencies of isolated alleles in the gene pool. But isolated alleles have little to do with phenotypic complexity and change. A broad interdisciplinary approach is required to understand how generative phenotypes generate ecological phenotypic gestalts. Linking generative phenotypes to genetic determinants requires new ways to visualize regulation of gene expression during embryonic development. The ways that the genotypic gestalt does change is the subject of active and fruitful recent research. Some of this research is pursued in specific model systems that help to illustrate aspects of both evo-devo that are not easily studied in fruit flies. Here we'll focus on 3 powerful model systems that have been especially illuminating: fruit flies, butterfly wings, and zebrafish. Although fruit flies have provided some of the earliest and deepest insights into evo-devo, other model systems are sometimes better suited to explore particular aspects of the evolution of development. For comparing the genetic determinants of visible patterns that play important roles in survival and reproduction among different species, butterfly wings have proven quite useful, as described in the following section. Zebrafish scales provide a model system for exploring cellular and molecular determinants of the phenotypic gestalt. Nusselin Volhard, who has served as director of the Max Planck Institute for Developmental Biology in Tübingen and leader of its genetic department, knew, along with her colleagues, that studies of invertebrate fruit fly development could only go so far in describing the ways that generative toolkits are deployed in vertebrates. Not long after they published studies covering several major aspects of embryonic fruit fly development, they turned their considerable talents and hard work to laying the groundwork for extensive use of zebrafish as a model system for the molecular genetics of vertebrate development. Like the fruit fly, but to a lesser extent, zebrafish offered ability to maintain strains with various key mutations and to visualize development in situ. Thanks to deep homology throughout the animal kingdom, it was not difficult to find homologous DNA, RNA, and protein sequences shared by fruit flies and fish, even though these toolkits are often deployed in different ways that generate distinct phenotypes. The molecular determinants of the 3D phenotypic gestalt in zebrafish scales. Most 3D cellular phenotypes are gestalts that are determined by several different generative tools, acting in a concerted fashion. In recent years, it has become possible to bring several lines of evidence from many different groups together to describe the regulation of an ancient signaling network that is activated during zebrafish scale development. A 2018 preprint by Aman, Fulbright, and Paraschi describes core features of this process by using a modern tool called in toto live imaging. Briefly, scale development requires the concerted activity of Wnt beta-catenin, ectodisplacin, EDA, and signaling by fibroblast growth factor, FGF. All of these genes and gene products play widespread roles in animal development. In this case, these genetic elements form a regulatory module that coordinates collective cell migration. In turn, this cell migration depends on another widely used genetic element known as hedgehog, HH. The authors suggest that a single ancient system has been adapted in various ways and is responsible for patterning of a variety of other vertebrate skin appendages, such as feathers, hairs, and ancient types of scales found in fossilized tetrapods, four-legged creatures.